and luciferase, along with additional autoinducer messengers to be released from the cell. This additional production of autoinducer messengers stimulates another round of protein synthesis, and the positive feedback loop results in rapid production of light-producing proteins. When these proteins reach a threshold, they react with each other in synchrony to liberate a beautiful eerie blue glow of the sea. Ta-da! The squid use this light to match the moonlight shining down from water and make their shadows invisible to predators when they hunt. This behavior of bioluminescence only works if my siblings and I work collectively. I can't do all this alone. In the past decades, scientists have found that all my relatives behave this way. We make signals, recognize words, turn on group behaviors that are successful only when all of us are working together. This process, called quorum sensing, calls for exquisite specificity. Only certain molecules will fit into specific receptors. Aside from bioluminescence, quorum sensing can also be used for secretion of virulence factors, biofilm formation, sporulation, and pigment production. You must be bored with my family history by now, right? Want to meet some of my relatives? My cousins, the myxobacteria, use quorum sensing for spore formation. When water or nutrients are in short supply, my cousins excrete a signal called factor A. Once the density of factor A passes a certain threshold, it warns the bacteria that they are in danger of starving and desiccation. My scared cousins clump together in groups of more than 100,000 individuals, forming a spherical mound called a fruiting body that can be 1/10 of a millimeter high. They then coat themselves with another message, factor C, to indicate that they are in correct position and density. Factor C triggers spore-forming genes that allow the bacteria to become spores, thick shelled spheres that can resist heat, desiccation, and starvation. My myxobacteria cousins can arrange in very complex shapes, pushing out through rotting wood and vegetation into the air to let wind, water, and even a passing animal help disperse the spores. A cascade of molecular signals are involved in spore formation. Remember my evil relatives I introduced to you earlier? Well, they too use quorum sensing in this case to initiate pathogenicity. They sneak into your bodies, grow, and when they have enough in number, they can do serious damage.
What did you do to him?
Just some laughing gas. Look, nothing up my sleeves. Nothing that won't put a smile on your face. Say cheese.
In the early 90s, papers were published describing cell-to-cell signaling in Agrobacterium tumefactions, which cause gall tumors in plants. In Arwenia carotovia, the architect of soft rotten carrots, and in a nasty bug called Pseudomonas aeruginosa, which accounts for 10% of all infections contracted in hospitals. Often deadly for cystic fibrosis patients and burn patients, P. aeruginosa makes itself impervious to antibiotics by surrounding itself with a biofilm that is mobilized by molecular signals. The staph microbes responsible for toxic shock send out molecular signals in order to compete against nearby staph colonies, disabling their rivals' quorum sensing circuits before becoming virulent themselves. Indeed, understanding how my wicked cousins communicate may definitely help prevent disease. Quorum sensing is not simply a black and white system. There are two general types of bacterial quorum sensing systems: gram-negative LuxIR circuits and gram-positive oligopeptide two-component circuits. Gram-negative bacteria have proteins homologous to LuxI and LuxR proteins of my family. LuxI proteins catalyze the synthesis of acyl homoserone lactone, autoinducer that freely diffuses into and out of the cell and increases in concentration in proportion to cell population density. The signal can be perceived externally through an external binding protein intermediate that presents the signal to the transmembrane receptor, or by directly interacting with the transmembrane receptor. The signal is then transduced within the cell. Also, the signal can be passively diffused through the membrane and interact with an intracellular receptor that then initiates a response in the cell. When LuxR binds to the AHL autoinducer, this complex activates the transcription of target genes by binding to DNA sequences at quorum-sensing regulated promoters. AHL inducers all share a common homoseron lactone and differ only in their acyl side chains. Even so, autoinducer for each family is specific because of substrate specificity of the LuxI proteins and the binding specificity of the LuxR proteins to the AHLs. In reality, it can get quite complicated. When the specific signal is not present in Vibrio harvaria, receptors initiate LuxU, LuxO phosphorylase, resulting in elevated levels of the QRR regulatory RNAs that block LuxR translation and prevent activation of Lux genes and other target functions. Signal perception reverses the phosphate flow, limiting QRR expression and allowing LuxR activation of target genes. Gram-positive bacteria use modified oligopeptides as autoinducers, often referred to as autoinducing peptides. IAPs interact with external domains of membrane-bound sensor kinase proteins to stimulate kinase activity, resulting in phosphorylation of the response regulator protein. These proteins bind to DNA and alter transcription target genes. AIP synthesis is very accurate because precise signal generation is guaranteed by the specific DNA sequence encoding the precursor protein. Some of my relatives are unable to synthesize their own signal and are capable of responding to signals of other bacteria, eavesdropping on their competitors, or synchronizing with collaborators. Many of my relatives inhabit complex environments and experience many different signals, some quite similar to their own, and so they need to discriminate between true signals and background noise. In addition, many bacteria recognize and respond to multiple chemically distinct signals that can be integrated to amplify expression of a particular gene or independently control transcription of a discrete set of genes. For example, P. aeruginosa has two AHL quorum sensing systems, LAS-IR and RH1-IR. Genes in the LAS-IR system are expressed before those in the RH1-IR system to allow the bacterium to express different virulence factors at various stages in the infection process. Interference is yet another possibility. Microbes and host organisms can release signaling inhibitors or actively degrade bacterial signal molecules. Several natural products interfere with our signal perception, including halogenated furanone compounds that inhibit AHL-based quorum sensing and inhibitory oligopeptides that block the function of closely related oligopeptide signaling systems. AHL degradation can be catalyzed by AHL acylases that cleave the acyl chain to produce homoseron lactone and fatty acids, and by AHL lactonases that cleave the lactone ring. Researchers are trying to learn more about regulatory proteins that transduce their response during cell-to-cell communication, such as transcription factors that bind to the DNA. For example, the TRA-R protein from Agrobacterium tumefacens and the P. aragonose LAS-R protein form dimers with each promoter bound extremely tightly to a single 3-oxo-C12HSL molecule and activate the expression of many different promoters, thus regulating many different target genes. Also, ESAR, found in Patina sowortii, a pathogen of maize, represses expression of RCSA, encoding an inactivator of CAPS genes required for exopolysaccharide production. A signal can inactivate ESAR, releasing it from its DNA binding site and reversing the effect. In addition to the continuous chatter among different bacterial species, signals are clearly exchanged with or intercepted by eukaryotic hosts. Several different plants are known to produce quorum-sensing mimics to manipulate microbial rhizosphere populations. You must be wondering how my parents distinguish between self and other. How do we count how many of me and how many of you and decide our next action, depending on who's in the minority and who's in the majority of a given population? How do we know when it's favorable to initiate a response?